The history of blueberry breeding goes back to 100 years ago, and then we have 85 years of that.
It was a collaborative program between North Carolina State College, it was at that time, and the United States Department of Agriculture at Beltsville. And the industry that developed in the state was a result of varieties developed for North Carolina because of one disease, a disease called stem canker.
There are not many public blueberry breeding programs in the country. So when I joined NC State, my goal was to shorten that period of breeding from 15 and 20 years to 10 years or maybe 7 years by marker-assisted selection. So you do the bee job, the thing that bees do, you do it manually, but in a specific manner and in a targeted manner. Shortening the breeding program has a large impact in terms of replacing the cultivars very faster. And in order to be competitive in the country and around the world, we need to be proactively continue this program and then use the new technologies to speed up this process to replace our cultivars one after another, so our growers can compete with other states. One of the traits that is important is fairness. So we are targeting to improve the quality of blueberries in terms of their shelf life and fairness. We had this farm for our program as long as I remember. We have our mechanical harvest trials here.
Machine harvested berries need to be a little more firmer and crisp so that they don't get all mushy when they're going through this process. It's a little harsh on the berries versus hand picking. So the growers in North Carolina, they really want something that's machine harvestable, so this helps us simulate the blueberry harvesting that they do, but in controlled settings in our fields. It harvests a lot faster. You only need a couple of crew people to be on it to help load the berries on and off and to run the tractor.
He's trying to keep up the variety breeding work and do all this basic research, which is absolutely essential. I mean, it's great. I would have, if I had been involved in hiring someone in the position after I retired, he's exactly the type of individual I've been looking for.
It's really an honor to be to inherit all of this heritage and just take it from here and then improve it.
Good morning. Good morning to everybody, and welcome to today's seminar. We are trying something new today. We are live here at Kilgore Hall, which is the home of our Horticultural Science Department at NC State, and we are broadcasting to you too. So I hope everything goes well. We have the honor of having Professor Dr. Hamid Ashrafi with us. Hamid got his PhD from Penn State University in 2007 in plant breeding and genetics. Then he joined UC Davis as a senior scientist as a postdoc, and then as a senior scientist in bioinformatics. Then in 2015 he joined NC State as a blueberry breeder. And that's the topic of his conversation. The blueberry breeding program at our university is more than 70 years old, and Hamid is using genomics and transcriptomic to study genes involved in different biological processes, including flowering, fruit ripening, biotic and abiotic stresses, and fruit quality. He's also teaching 2 courses, one undergraduate course in small fruit production and a graduate level course in breeding with genomics. Hamid, it's an honor to have you here next to me, actually. I'm going to stop sharing and give you the floor to you. I'm going to mute myself.
Thank you so much, Carlos, for your introduction. So before I start, I just need to share my window, make sure that we are good to go. Okay, everybody can hear me in Zoom everywhere? Okay. Hello, everyone in Zoom land and here in the room. Thanks for attending in person. So the title of my talk today is Sustaining the Supply of High-Quality Blueberries by Leveraging Applied Breeding, Genetics, and Genomics. So I'm focusing on breeding part of the program more today than the genomics part, but because this is an international series, and I think there are so many people who may not be familiar with blueberry or from other countries that blueberry is not a thing over there. I've been in several African countries that they haven't seen blueberries. They don't know even what blueberry is. So I'm going to start from the basics and go from there. This is what blueberry look like. And let's see if I can forward. First of all, I would like to acknowledge my team at NC State, the former team members at the bottom of the screen. And I need to change this to pointer. Okay, there you go. So these are my old people that they just left the lab, but the top part is the current lab members: Jessica, Jenny, Felicia, Nisa, Gray, Kristen, and Ellen are currently working in the lab. And also I would like to thank the funders of the program. Blueberry Council is one of the largest supporter of our program. NCDA, USDA, NEFA, NSF, the Southern Region Smart Food Consortium, Blueberry, not Blueberry, Plant Breeding Consortium at NC State University also support my program every year. And in the past, we have received funding from Plant for Human Health Institute as well. So with that, I would like to proceed. Our research are communicated through our website and Facebook and our blogs website. The website, if they are long to remember, you just type in hamidashrafi.com and it goes to the Blueberry Genetics, which is at cals.ncsu.edu. So that domain is redirecting you to my lab website. So where we are, this is the map of United States. And as you can see, we are in the East Coast and the Mid-Atlantic region, or sub-Mid-Atlantic region. And this is a zoom out our state, North Carolina. And here is where we are located in the Wade County in Raleigh. And our farm is located about 2 hours south of here, near the coast in New Hanover County. The yellow circle shows the regions of North Carolina or counties that blueberries are growing. It's Bladen County, Columbus, Penders, Onslow, Dauphin, and Sampson. So these are the major blueberry growing regions in North Carolina. We have 2 stations in Moore County and Cumberland County. And so those are other research stations that we use for our program. But the main station is located in here in New Hanover County and Castle Hayne. So let's switch the gear and talk about blueberry. What is blueberry and where it's come from? Blueberry belongs to Ericaceae family. The genus is Vaccinium. It has nothing to do with cow, but when Linnaeus called it Vaccinium, people don't know why, but it is Vaccinium. So it has 22 sections, and then the main section that blueberry comes from is from Cynococcus. And within this section, we have about 17 species. The main species that we are focusing today are Darrowi. Vaccinium darrowi is a diploid blueberry. Vaccinium corimbosum is what you buy from the market. It's a tetraploid blueberry. If you go to state of Maine, and then you find blueberries in the market over there, sometime in August, those are low bush blueberries from a different species. Again, it is tetraploid. It's called Vaccinium angustifolium. And then if you go south toward Florida and Texas, there is another species. It's a hexaploid, and it's called Vaccinium virgatum. Formerly, it was called Ashii. These are different pictures of blueberry in different seasons: winter, spring, summer, fall, and summer and fall. I'm just focusing on winter. You see that it's a deciduous plant, so the leaves fall, and at this time you see the flower buds that form from the last season. Spring, it starts with beautiful pinkish flowers, and then these pink flowers eventually become white. And some varieties they produce flower before leafing, and some they leaf before flowering. So that depends on the variety. During the summer, you have the chance to pick up the fruit, and that's the cluster of the fruit on the shrub. And this is the fall picture where the leaves start to change color, and some of them they have beautiful color, fall colors that can be used for ornamentals in some regions. And as you can see, the flower buds for next year fruit are forming in the fall. Blueberry in North Carolina is about $67 million business. And each year, 46 million pounds are produced in North Carolina. Depends on if it's a good year. We are sixth in the country in the U.S. in terms of production. Sometimes we are neck and neck with California. Sometimes we are 7, they are 6, and vice versa. There are about 10,000 acres. If you are international, that translates to 400 hectares. 10,000 acres. I know it is 4,000. Oh yeah, you're right. 4,000 hectares, and so it accounts for 88% of the total production of the U.S. And about we have 100+ number of operations in North Carolina. Some operations are big; they are about 500 acres, 200 acres, 300, and some of them are small farms and pick your own. If you look at the market and the whole world and where we are located or where we are situated, United States back in 2018 was the top producer of blueberries in the world. Starting 2020 and now the market has shifted toward Peru and China. So those are the big players now, and our production has been steady, hasn't been increased or decreased. Some years have been decreased because of the weather. But as you can see, China and Peru have grown so much during the past 3-4 years. Chile has kind of constant production, has lowered this past year. Mexico is growing. Poland for European market is growing. And if you can look at the trend all over, it's growing. It's because of the health benefits of blueberry, and people are more conscious about eating blueberries. So which regions are the leading blueberry market? North America. Of course, United States was the first country, but as you could see by the numbers, we are the third now. Canada, Mexico in the North America. In Europe, Poland, Germany, France, UK, Italy, and Russia are the major producers. Balkan countries are also coming to the play recently. Asian Pacific countries, China is the major producer, and Japan, South Korea, India, Australia, Indonesia, Thailand, and Malaysia are getting into the market. In Latin America or South America, Chile, Peru, Brazil, Argentina, Colombia, and other countries are also producing blueberries, but it's not as significant as other countries that I've listed here. In Middle East and Africa, Turkey and Morocco are becoming major players for European market. Similar to US market that we have year-round blueberries, that are blueberries. When we don't have blueberries, they come from Chile, Peru, and South America and Mexico. Turkey and Morocco are suppliers of blueberries off-season for Europe. So how do we culture and management of blueberry, how do we do that? Pollination is a requirement for having the fruit. In our region, we have a local or native bees that are doing the pollinations for us. As you can see on this picture, this bee look like a bumblebee, but it's a little bit smaller. it makes a slit on the side of the flower, and then it suck up the nectar from that slit that it makes. And then as a result, it pollinates without knowing what it's doing. But so this is a major thing for good production, bigger fruit. Blueberries like to be cross-pollinated. Some varieties are more self-pollinated than others. But overall, if you cross-pollinate them, you get a better fruit and better fruit set. Pruning is an annual work. You have to prune them every year, removal of old and weak canes, shaping and training the trees or shrubs. Irrigation in our region is, it has a dual purpose. If we have a drought, like duration, we can use our overhead sprinkles and water the plants, and then we can use it as a mean for frost protection. So for us, it has dual purpose, but in some other countries that they produce blueberries in pots and substrate, it's the main source of water for the plant. Because in North Carolina, we plant the blueberries in the soil. We don't plant them in the substrate. Fertilizers and organic matters are important. We have a type of soil in North Carolina that is called North Carolina clay or sandy clay. Sand, sorry, not clay. And this sand, underneath of this sand is a layer of organic matter. So it's a perfect soil for blueberries. When the root gets to that layer of organic matter, it's very acidic and it provides nitrogen that it needs for good growth. Pine bark is amended in some regions that they don't have that organic matter, and pine bark helps to acidify the soil, and for growth of the blueberry, you can even plant them in pine bark. Different blueberry types, I talked about them. Rabbit eyes are the hexaploid. They don't have a good, I would say, they have a taste, but it's they have a thick skin that it's not preferable by consumers. These are the blueberries that can grow in low chill regions, and I'm talking about the chilling hours and what it is important for. We can grow northern blueberries here, but the flowering time is going to be postponed because they don't receive number of hours, chilling hours that they need. Southern high bush blueberries are a type of blueberries that develop in mid-50s, and these are a cross between native southern blueberries with the northern high bush blueberries. We have a couple of pentaploids that are seedless blueberries. Robinson and Heintuga are two examples of those. And those are crosses between tetraploids and hexaploids. What is chill hour? Chill hours are hours below 7 degrees Celsius or 45 degrees Fahrenheit required to leaf and flower. And it happens here usually between October to February in North Carolina. As I said, we need to have soil, acidic soil with high % of humic matter, including organic matter, and in the USDA hardiness zone, we are zone 7 to 8. So this map shows a rough shape of the chilling hours per latitude in the US. Florida has 0 chill hours, as you can see. And then as you go up to Michigan and Pennsylvania, up north, you get to 1,200 chilling hours. And you need to accumulate these chilling hours for the good flower set and fruit set, flower bud set. And the zones are on the right-hand side. If you are not familiar with the map of US, the zones start from north to south, and then the numbers increase from 1 to 10. And as you can see, we are in North Carolina, we are in zone 7 to 8. High bush blueberries, they require about 800 to 1,200 plus hours. Rabbit eyes require 400 to 800 plus hours. And southern high bush blueberries, they can have 0 to 900 hours. And those are on the right-hand side column. You see the hardiness zone of the blueberries in the US. When I talk to growers, I ask them, pick research and choose your own, plant as many as varieties you want, and then see which one works better for you. Our job here at NC State Blueberry Breeding Program is to provide them with the high quality blueberries that are adaptable to North Carolina. But our growers also try other varieties from other states and other breeding programs as well. So the good blueberries are the ones for growers that they don't stem. When you pick it, you don't want to have a stem. And then for mechanical harvest, if the stems don't attach, that cause a problem. Cracking is a problem. If blueberry cracks during the rainy season, it's not a good variety. When you pick it, if you have a stem scar that is wet, that's not good for packaging, and too soft is not good for shipping. Poor flavor. If you buy a blueberry and you don't like it, you don't go back and buy it again, that brand. So it's very important for them when they put their brand on the blueberry and for the consumer acceptance. And next time that you go there, if you see that brand, you buy it again or you don't buy it again. Susceptibility to diseases, that's another thing to consider when you grow blueberries. And then color too dark, it's not good. Consumers, they like to have whitish to blueberries. And size, of course, is a major thing. If blueberries are too small, consumers don't like. They like large blueberries. At least as large as a quarter is favorable. So, and these are the other things are considered for the production. Poor bush survival, if it lasts only a couple of years and then dies, so it's not good. If it ripens too late, sometimes you miss the window of having the product in the market, and then it gets too cheap or not profitable for the growers to sell it. But if it is too late, sometimes it's good because nobody has blueberries, then the price goes up. So it depends on the market. They usually have, I'm going to talk about that process that or the practice that they do to have an overlap of blueberries over the season, longer season. Of course, yield is a big thing. If the yield is not good, our growers, they remove the varieties, they replace them immediately with a high yield variety. And the last thing is mechanical harvest, which is very important for blueberry. Nowadays, that the labor is a problem in the US, we are looking for mechanically harvested blueberries. As I said, color of blueberries is very important. On the left-hand side, you see Pinnacle, and then on the right-hand side is Biloxi. Biloxi, a few years ago, it was covering about 90% of the market in Mexico. So when it comes, these two blueberries were picked at the same time on the same date, and after 2 hours, the one on the left-hand side is blue and nice. And then after 2 hours, Biloxi has lost all the bloom that is on it. The waxy bloom on the fruit helps the fruit to become to have a bigger or longer shelf life, and it looks more attractive to consumers. So this is very important for shelf life and for shipping. So what you get usually in the market is the right-hand side rather than the left-hand side picture. We also have we used to have a researcher in our station who was planting all the varieties that we're introducing each year by different companies, by different entities, and he was testing them in North Carolina to see which one is more adaptable. But we also do the flavor testing. So we invite our growers here, and then we ask them to taste our advanced selections and current varieties. This is important for making decisions about good flavor blueberries. This table shows the firmness of blueberries. The firmness is one of the main characteristics for shelf life for mechanical harvest trial or harvest picking of blueberries. And you can see that the variation is from 150 to 228, or even Suzy Blue is 243. And the higher this number, the better it is for blueberry. This table shows the ripening time from May to June, and the ones on the left-hand side are the early ones. You can pick on May 19, for example, 44% of the fruit is ripened. On the right-hand side, we have at the bottom of the table, we have some of the high bush blueberries in North Carolina, and as you can see, high bush blueberries here, they are ripening very late. Even in June 15, we have 5% ripened Aurora and Liberty here. So that's the way that our growers usually extend their season from May to June, and now they are pushing for even more to July and August. So this shows how the varieties are overlapped for the southern high bush blueberries from the early one to the latest. Some blueberries are finishing from start to end. It's maybe 10 days. And some of them, they can go even for 2-3 weeks. So each number here shows the weeks. And then the bigger the bar means that you can pick these blueberries over a larger time span. This shows this northern high bush blueberries like Duke. Duke is the earliest of all, and Elliott is the latest of all. So depends on, as I said, our growers are mix and match the varieties to have a good overlap of the season. So blueberries, I'm going to switch to gear and show you the quick rundown of the blueberry process and how do we do the breeding. So if you can see in the right-hand side picture, our pedigrees are very complex from left-hand side to the right-hand side where you have some advanced selections. You see that we have incorporated a lot of species. Each color of these boxes shows a different species. And eventually you see that the corimbosome is increasing. The light blue color is corimbosome. But if you go back several generations, you see how complex are our pedigrees. And these are thanks to Dr. Ballington, my predecessor, who was enhancing germplasm, and he was doing a lot of expeditions, collecting these germplasm, bringing them here, and then crossing them with corimbosome. So when it comes to breeding, you have all of these species to choose from. Some of them are easier to cross with, like the one from Cyanococcus are easier to cross with Cyanococcus. But if you go across different sections, it's getting harder to make the crosses. You still can make crosses, but the fertility is various. So when I joined NC State, I decided to develop SNP markers for molecular breeding, and we collected a lot of species from Darrowii, Arboreum, Rabbidis, Corimbosome, Angustifolium, and you name it. So these are some examples of the species that we collected at the beginning. Darrowii, Eliotii, they have different shapes and sizes. Pallidum, and Angustifolium is the one that is grown in Maine, and this is the way that they harvest Angustifolium in the state of Maine. This picture shows the breeding process of blueberry. Minimum 15 years is required. I have adapted this from University of Florida, but this is not what we usually do. We have our own scheme of breeding, but it's more or less the same. As everything starts from a cross in the first year, and then the selection in subsequent years, and then multi-site selections in later stages, and then choosing the best of the best. We usually make 100 crosses per year. from each cross, we plant 100 seeds. So we have 10,000 seedlings. And since it's a continuous process, and then we do this every year, we're gonna have overlap. And by the year 15, you have the things that you are starting for the next 15 years. So what I'm working on, they have been developed in 80s and 90s. And in University of Florida, they usually have 20,000 or 10,000 seedlings per year. The first, because they are doing high density breeding. Then the second year or the second stage, they triage that to 1,000. And then in the later stages, they go to 110. So in each stage, you reduce or you put more pressure on the selection. In our program, we don't have the capacity to do that. We do it differently. From 10,000, we go directly to 100. So we don't do the intermediate selection. And then from 100, we go to 10, and then 1 or 2 for cultivar identification or naming. So this is just a quick show of how we do the breeding. So that's the maternal parents. We select them in the field, we dig them out, we put them in the pots, and we bring them to the greenhouse. We put them in the cooler to chill them and to synchronize the flowering. We usually bring the male parents out of the cooler first, and then the female parents a second. We collect pollen from the male ones, because we bring them out of the cooler a few weeks earlier. This picture shows the structure of the flower, and with the petals removed, and then you see the pistil and also the stamens. Once the male parents are ready, we extract pollen from them by this procedure that you see. We just roll them between our fingers and then collect the pollen in these tubes. And then these are the bud stages. These stages are too early for emasculation for the female parent. This stage on the left-hand side is the right stage for emasculation. We emasculate the flower at this stage, and then we remove the flowers that are already open because they have been exposed to maybe outside pollen, so we don't want to have those. And then we keep a few of the flowers that have been emasculated on the flower on the bush. We usually do 150 flowers per plant or per cross. And this is what it looks like after emasculation. You have a stigma just exposed. And then once we have collected the pollen, we can have the fresh pollen or frozen pollen, and then we use these glass rods to do the pollination the day after emasculation. So after emasculation is done, after pollination is done, the fruit starts to develop, and you can see by looking at the stamen and by the style, and it usually turns up toward the sky or toward the ceiling, and that indicates that you have had a successful cross. We let the fruit to develop and ripe, and then this is the ripe F1 from a cross, a hybrid between two varieties or two species. And then we extract the seed with a blender. We collect all of these seeds into a bag as they ripe, and then we keep them in the fridge until everything is ripe and picked. Then we use a blender to blend them, and then we extract the seed this way. I have put 2 videos here for showing audience if they want to learn how to extract seeds from blueberries. So once the seeds are extracted, you can keep them in the cooler for a few weeks, or you can plant them or sow them fresh. So this is the way that you, on the right-hand side, we put the seed on the top of the soil. We don't cover it with anything, and then we put it under the mist until the seeds are germinated. After 30 days, it takes a long time to germinate. After germination, we transplant. Once they are 1 inch tall, we move them or transplant them to these single plugs. And then after a year, so this process usually starts in March, April. We collect the seed in August, and we sow the seeds in November. So this picture that you see is probably January of the next year. And then this is the next November. So it takes 1 year from seed to seed to this stage, to get to this stage. And then we take it to the farm, and then we use a tobacco planter to plant it. These are 1-year-old seedlings, 2-year-old, and then 3. So as you can see, they grow very fast in this type of soil that we have. And that's the type of soil that I was talking about. It is covered with sand, and underneath there is a layer of organic matter. We start evaluating these on the 4th year or maybe 3rd year, and sometimes the situation is on the left. You see the situation is what you see on the left-hand side picture. We have to have boots, and then it's because of the rain and the type of soil that we have. Although it is sandy, but because of that layer of organic matter, the drainage is important. So sometimes it doesn't drain very well. So we do the evaluations. This is an evaluation form. I'm guessing that all of the audience or most of the audience here are breeders, or they want to be breeders. So we have these kind of forms. We walk through all of these 10,000 seedlings that we created 4 years ago, and then we evaluate them based on the crop. Means if it has a good productivity, we check them one by one to see if they are firm, soft, medium. We eat them one by one to see what is the flavor. And in a given day, you can imagine how many blueberries we eat per day when you walk through 10,000 seedlings. And they are, each of them are different from another one. It's like human children. So each seedling is different from another one. It's different from other crops that you make an F1 and all the F1s are look alike. But in blueberries, each F1 is like a human child. So after we make the selection from 10,000, we go to 1,000. We label them, and then at the end of the season, sometime in August, we go and get these softwood cuttings. And then softwood cuttings are the best type of cuttings for propagation of blueberries. And then we put them in the pine bark, and then we propagate them from 1 plant to 24 usually. And then those propagated plants go to 3 plant plot observation plots for 5 years, and we do it in a replicated trial. If we have more extra plants, we send it to other locations. We do multi-location trials as well. During this time, we collect a lot of data, including yield, berry size, berry firmness, berry color, stem scar evaluations, fruit blemishes, post-harvest storage, and sugar and acids. So these are the things that we do in the lab with different instruments for acidity, for fruit firmness. We also talked about mechanical harvest and what our growers are asking us to do. We put a subset of these selections in mechanical harvest trial and replicated trials, and we harvest them by mechanical harvester. This mechanical harvester simulates what the growers do. It's not exactly what they have, but it is a smaller version of the growers' mechanical harvester. Of course, theirs is much better and more fancier than this. So after collecting data for mechanical harvest, we do also flavor testing, and then we call them advanced selections. So this is one of our advanced selections. Our philosophy of breeding is to develop early, mid, late blueberries. So this advanced selection 5288 is one of our in pipeline that is ready or close to be ready to be called a variety. And it's an early season blueberry. So 5300 is another selections. I'm just showing you some of our latest selections. 5289, and on the left-hand side, you see the shape of the fruit and then the shape of the plant on the right-hand side. And this one is mid-season. I don't have anything for late season to show you, but we are working on it, and then we are selecting for late varieties. As of last year, we have started to select the late ones. 5303 is another mid-season, early to mid. And then 5305, all of these 53 series are very good quality blueberries. We hope that we name them and introduce them soon. One of our newest releases is 3104 or Rowan. This variety is a replacement for New Hanover. It doesn't have the problems that New Hanover had with the stem scar, and it has the same flavor and the same productivity. It is a sister line to New Hanover, so they have the same parents, but we decided to name it as Rowan. Rowan is one of the counties in North Carolina. By the way, most of our varieties are named after counties in North Carolina. We are also working for home growers and develop blueberries for ornamental that can be used as ornamental. This is a new release called Splendid Blue, and it's NC 4499, and it has a good stature and form for home growers. So you can prune this, and then you can treat this as a bush in your backyard. And it has a nice glaucous leaf and bluish color, and it produces decent sized fruit. So if birds allow you to have fruits in your backyard, this is a good variety. Then it comes to commercialization and patent. So this is where all the blueberries are propagated in nurseries. So blueberries have to have ability to be propagated in tissue culture. If at the end of the season or at the end of the cycle, you have a variety that's super duper, but it cannot be propagated tissue culturally, it's not useful because it doesn't get to the inroads and the nurseries. So that's the way that the nurseries produce thousands of or millions of plants. So this is an example of a nursery in Florida that I visited a few years ago. And this is tremendous amount of number of plants that you see in this picture. Okay, how we can speed up the process of breeding by molecular markers. We are still working on this aspect of our breeding program. One of the things that you can do is increase the number of generations per year. Instead of one time crossing, just do it 2 times. Sometimes if you get the softwood cuttings and then you root that softwood cutting, you can get the flower on the first year. You can back cross it to the other parent if you want to. So this way you don't need to wait 3-4 years for the flower to develop. You can just do it on the first year of softwood cutting. You can do disease screening of the seedlings if you have markers, and then selecting the seedlings in the year 2 instead of year 3 is another way of selection. So that's where the molecular markers can help, that you can do early generation selections and do marker-assisted breeding and genomic selections in early generations rather than waiting for 15 years. So as I said, we decided to develop markers for our program. And one of the type of markers that we developed was SNPs, or single nucleotide polymorphisms. So these are abundant number of markers in the genome. Everybody knows there are millions of SNPs scattered around each genome. Another aspect of the work that we are working is genome editing and transformation. And so I'm not going to talk about it today, but it's an ongoing work by Felicia, one of my PhD students. And so where we are right now with the genomics, I think University of Florida is more advanced than us in applying genomics and genomic selections into their program because of the funding, and that program is well funded versus our program. But we are getting close. So what we can do with these markers, we can use them in phylogenetic studies, in trait identification, in DNA fingerprinting, parental testing, identification and location of quantitative traits, or QTL mapping, and marker-assisted selections, or genomic selection. So when we started this process, we had 29 vaccinium species, 29 vaccinium from 18 species. We resequenced them, and then we put it in a pipeline to detect the SNPs. And then, as you can see here on the right-hand side, all of the Cyanococcus group, they were grouped together. And then these are other sections of blueberries from these three that are on the left-hand side. So then in the next phase of this process, we increased the number of species to 30 and number of accessions to 70. And then we did more. We developed we converted some of these SNP markers to Cas markers that can be done in the lab with the PCR, or converted them to PCR-based markers. We also developed a new tree based on all the new species that were added to that previous tree. And now we know where they are related and how they evolved from each other. The other use of markers that we use in our program is for patent and IP protection. So if our varieties go to somewhere and somebody get cuttings of it, we can do a CSI project and then find who did it or how they did it and if it is our variety or not. So using 25 markers that from the CASP's markers that we developed previously, we selected 25 markers that can detect these varieties wherever they go. So for example, this NC4499 that I showed you previously, we run the markers and then we have the fingerprints for those, and then we're going to put it in the patent application. When we did it with the parents of that, it's really nicely shows that the pedigree is correct. And these two are the Sunshine Balloon and 48 and 3048 are the two parents of 4499, and 4499 is just between Sunshine Blue and 3048. So it fell behind it right where it has to be. So we had about 1.7 million SNPs. From this set, we also selected 60,000 SNPs. And then we had a diversity panel of 260 genotypes of blueberry from Corvallis repository and also our germplasm. We phenotyped these 260 genotypes and we for 2 years, and then we genotyped them with those 60,000 markers. For the phenotypes, we did it field evaluations for fruit ripening, flowering time, and all the phenologic traits. And in the lab, we did colorimetric measures, fruit weight, pH, acidity, firmness, and whatnot, and cyanin contents. So from that 60,000 SNPs, we developed baits, and then we captured those segments of the DNA of these 260 individuals, and these are the SNPs that you see in our diversity panel. And then we put it in a GWAS program that has developed for polyploids. And then we detected some SNPs that are potentially associated in different chromosomes with for example, here is with firmness, and then with fruit weight, which is a very important trait for fruit size, actually. So where we are going with this, quantitative characterized a large range of fruit and phenotypic characteristics. That's what we did. We detected hundreds of putative genes and isolated candidate genes. We have a list of candidate genes for our blueberries right now that we can explore further to see their function. And so this study is the foundation of marker development for marker-assisted selection. And with that, I open the floor for questions. And thank you so much for listening and being patient with me.
Thank you, Hamid. And we already have a couple of questions in the chat. I don't know if you can read there, but Jen is asking if you have implemented altering photoperiod to shorten the development time of flowers and fruit in early stages of your program. If you have play around to shorten the
not through genetic engineering.
no, just through induction.
Yeah, we have all the data. Do you know which accessions flower first, and then which accession flower last? And we have all of these data that has not been published. but through genetic engineering, a scientist in University of Michigan, they overexpress a flowering gene in blueberry, and they showed that by overexpressing that gene, you can have blueberries that are 1 year old that are full of flowers. But we haven't done it through, we are trying to use traditional breeding here.
Yeah, and in which stage of the breeding pipeline would you use genomic selection?
As I said, we haven't employed genomic selection yet. That's a question that you can ask Patricio Muniz in University of Florida. But I think they are using a stage 3. So if you go back to the picture that I showed you, they have a stage 1, a stage 2, a stage 3. They deploy genomic selection in stage 3 because it is a lot of resources and money requires to do in earlier stages because you are dealing with thousands or thousands of genotypes.
Yeah, the next question, are you working on genome-wide identification for this crop? I don't.
Oh, GWAS, I think that's the question, maybe. I think we did a GWAS study, and then we are expanding that GWAS study to our next phase of our breeding program. Right now, we are planning to do the GWAS on our advanced selections rather than the germplasms.
How does the higher ploidy levels impact the rate of successful fruit set?
So as the ploidy level increases, depends. For example, rabbit-eyed blueberries that are hexaploid, they are very productive. So increase, but if you go back to the diploids, they are also productive, but they have a smaller fruit. So ploidy has increased the size of the fruit, and I think it is a matter of cross-pollination rather than the ploidy level.
This is a question from me. Question from me, Amit. Have you started to look at parthenocarpy?
Some blueberries are producing fruit without pollination, and then we have seen it in our greenhouse because we don't pollinate there. There is no bee in our greenhouses, and some varieties like Legacy, if you don't pollinate them, the flowers die and you don't get any fruit. But we have a very new advanced selection is 5271. That number is amazing how it produces fruit without any pollination. So we are contrasting these two genotypes to see what is the difference and what is causing that to be more prone to parthenocarpic versus the other one that is not parthenocarpic. These are the two good genotypes to look at.
It says here, you mentioned about DNA fingerprinting to protect the IP for your varieties and determining 25 CAS markers for this purpose. How do you pick those markers?
So as I said, we had 1.7 million SNPs. We randomly across the genome selected about 144 of those SNPs. And then we tested all 144 SNP markers. And then out of those 144, we selected 25 that are segregating in a simplex fashion or a diploid fashion. So you can see the heterozygous, homozygous, and homozygous alternative allele similar to a diploid species.
Have you found markers that are associated with cold hardiness?
Another colleague of mine at USDA, Dr. Jeannie Rowland, she has been dedicated her life into cold hardiness. I think that's the right question to ask her. But she has published a couple of papers on QTLs for cold hardiness in wild blueberries, like diploid blueberries.
Do you believe that genetic engineering have less time in improving traits than conventional breeding? Will allow to spend less time in improving traits than conventional breeding?
So if you look at new varieties that are coming from China, you may rethink that you are doing everything wrong in the US. So I was looking at some patents that came from China and some varieties. The selection was made in 2009, and then variety was named in 2015. It's 6 years from selections to naming a variety took them 6 years. And I think the way that they do, they don't care about the parents. They just eat the blueberries, they extract the seed, or if it is a good blueberry, they get it, they get their hands on it, and then they plant the seed, and then they select among those. So they don't do any crossing. So I think that's the easiest way to breed for blueberries. If you like a blueberry, just plant the seed and then make selections among those. And then that way you probably can increase the chances of, improving it. That's the only way that you can speed up the process. If you want to do the path that we are taking, like having, this parent with this parent, that takes about 20 years.
You're planning to increase your germplasm collection, adding accessions from other countries.
Yeah, we are collaborating with some countries like Italy. They are taking our material, our advanced selections, and they are trialing there. We have collaborated with Global Plant Genetics to do the same thing. And we are expanding it to Morocco and Portugal, probably, and Turkey also, to take our advanced selections and test it in their environment.
I was going to ask you about that, like Peru being one of the countries I know you have visited and collaborated with them. At what stage of your programs, for example, would you send materials to Peru to be tested?
That's a strategic decision that our higher administration needs to take, and our growers. If they allow me to do that, I will be open to do that. And I think with nowadays companies, I didn't have time to talk about different breeding programs. I have another talk next week for Portugal blueberry growers that I'm talking about the status of breeding programs in the US. And companies are going everywhere. So Fall Creek has entities in Peru, Chile, Mexico, Spain, and Morocco, South Africa, you name it, China right now. And companies are doing it. And if we don't do it, we're going to fall behind. So we have to do it. We have to be open-minded and open the doors for South American to try our material.
How productive are the low chilling our varieties versus the ones that require more chilling?
In terms of productivity, it depends on the variety and it depends on the soil. So the production system that we have in North Carolina is different from other places. If you go to Europe and Morocco or even in the US, if you go to California, the production system is different. So, and the productivity is a balance of number of shoots that you leave and the number of flower buds that you have from the last year, the pruning practices, and the fertilizers. So it depends. If you are asking me about North Carolina, we have varieties that are performing better than the past. And these advanced selections that I showed you, these are, they have the potential of being replacing other varieties. But I don't know how they perform if they go to substrate. So we are collaborating with our Italian counterparts to see, because they do it in substrate, we're going to see their performance in substrate. And we are looking for collaborators to take our material, put it into the substrate, and then test them in the substrate.
That's actually the next question.
Is what kind of collaborations you have in your program? You mentioned Italy.
We are public breeding program, and we are open to all type of collaborations, including germplasm exchange through MTAs, and if you have something that you want to test here, you are welcome to send it to here.
Sorry. Yes. Is there any questions?
Are there any questions here in the audience?
Yes,
Peter.
I have a couple of general questions about blueberry growing. How often do growers replant their fields?
How do growers replant the field? That's the question.
if they see that a variety doesn't perform well, they remove it in the 4th or 5th year. It's a big investment, But if they don't see any light at the end of the tunnel, or that variety was said it is good, but it doesn't perform in North Carolina, they remove it. But if it performs well, like we have varieties that have been lasted 20 years, like O'Neill, Rebel, these are the varieties that historically they perform very well. They peak at the production on the 5th to 10, 15 years, and then after 15 years, they go down. So if you have a plant that is 20 years old, I think it's time for a replacement.
You have to put netting over the fields, just like a overall.
No, in our location, we don't have much birds in Castlehane, but we have a lot of birds problem in Sand Hills. Over there, we have a bird deterrent system that mimics the sound of hawks, and so when we turn that machine on, it is very effective. We have seen the effect of that machine to deter the birds. Some people use falcons and use falconeries, so they come and use actual falcons in the farm. But this bird guard is a nice deterrent machine.
Well, thank you, Hamid. Let me see if I can unmute myself. Going up. Please go.
Yeah. Yeah, thank you, Hamid, for your nice presentation, and thank you for attending this seminar. Next Thursday, we'll have the presentation from Dr. Fekadu Dinsa from the World Vegetable Center in Arusha, Tanzania. And he's going to talk about African traditional vegetables, research and development in Sub-Saharan Africa. The photo you have there is amaranth. He does amaranth breeding among other species. So it's going to be a very interesting talk. Please join us at the same time, 11 a.m. Eastern time here in the US. Thank you so much for the people that joined us from odd hours like India, Pakistan. Thank you so much, and have a great weekend. Okay, that's it from here. Thank you.